Let's clap hands for this occasion. Thank you very much, colleagues. You can get seated. Today, colleagues, my name is Professor Thobeka Nganywa, the Director of Research and Innovation at WSU. Colleagues, today we are here on Prof. Niba's inaugural lecture. I know the feeling, Prof. It's quite a nice feeling to be inaugurated. I was also inaugurated in the same university. I know the feeling. We are sharing the same feeling today. So, colleagues, we are going to come and ask Professor Lillington to come and welcome us in this occasion. Over to you, Prof.
Moweni, good afternoon. Wamkelekile. I want to start by asking everyone to stand for a moment of silence in memory of the DVC Institutional Support and Development, Sinetembe Mbapani. Thank you. I would like to welcome our inaugural lecturer, or inaugural professor who's giving his lecture, Prof. Niba, and his wife, Mrs. Niba. Very welcome. I'd also like to recognize and welcome the DVC Research and Innovation Professor. I'd like to recognize and welcome the Executive Dean, Prof. Modi. I'm a bit short for here. I can't see the people in the front row. I would also like to welcome the Professor Curia, who's the academic respondent to the lecture. It is really an honor to have professors from other faculties. I know there are two professors from humanities here. Thank you very much. I'd like to welcome all the staff and the students here today, especially those from the Faculty of Natural Sciences. It's a wonderful day. But those who were interested in the topic and came anyway, a huge warm welcome to you. And I'd like to also recognize the Program Director, Prof. Nganywa. Thank you for doing this program. Our apologies for starting a bit late. I think that we are aware that there's a memorial in Butterworth, and as a result, things got a little bit delayed. And at the same time, offer the apology of the Vice-Chancellor, Prof. Sheila Sontra, who was couldn't be at two places at once, but sends her best wishes to everyone here. It's really a pleasure to welcome everyone to this lecture. My background is agriculture, so the topic is of great interest to me. And I looked at some of the titles, as well as some of your history and where you've been and the research you've done. And I'm really excited to actually listen to your lecture. Since we started late, I don't want to be long, but it is a warm wamkelekile to everyone. And please enjoy the lecture. I'm sure it's going to be entertaining. Even the respondent, I'm sure, is going to be of interest. And the lecture, I'm sure it's going to be entertaining. Even the respondent, I'm sure, is going to. Thank you.
Can we clap hands for Prof? Thank you very much, Prof. Lillington, for that warm welcome. I'm feeling warm already. Are you? Yes, thank you very much. Professor Feza, can you please come and tell us who is Professor Niba?
Good evening, colleagues. Prof, I feel a little bit taller because I'm on heels. I know I'm not taller than you. Today, colleagues, I am presenting to you Professor Augustine Sue Niba. Professor S.A. Niba was born in Buffet in the Mesam division of the Northwest Province of Cameroon in 1965 to the family union of the late Taniba Tafa and the late Mrs. Regina Manga Niba. He trained at the University of Calabar, obtaining a BSc honors in the Department of Biological Sciences in 1988. Prof. Niba received a Master of Science in Applied Entomology from the Department of Agricultural Biology, University of Ibadan in 1990. He obtained another Master of Science degree from the University of Natal in 2000, where he continued to PhD in entomology and conservation ecology, graduating in 2004 from the then University of KwaZulu-Natal. Prof. Niba joined Walter Sisulu University after a 2-year postdoctoral position at UKZN in 2006. He has been a full professor of entomology and conservation ecology since 2023. He served the Department of Biological and Environmental Sciences as program coordinator for the Pest Management Program. At the time, the Bachelor of Science degree and Diploma in Pest Management Program were offered within the zoology unit of the department, which was under the School of Applied and Environmental Sciences, which is now Department of Biological and Environmental Science. His key performance areas were teaching undergraduate and postgraduate students, research and community engagement. He has been actively serving Walter Sisulu University at the faculty level as a faculty member of Natural Sciences, chairperson of research and higher degrees. Since then, he has been tasked students, 60% of whom are females, and graduates have been recruited into Wits, Pretoria, Stellenbosch, and Rhodes universities to pursue their BSc honors, MSc, and PhD degree qualifications where they qualified. Some graduates have found jobs in various sectors of the economy, while others have joined the Walter Sisulu University workforce. Based in a historical disadvantaged university, Prof. Niba had consistently taken up the challenge of championing innovative and adaptive research in his capacity as chairperson of the Higher Degrees and Ethics Committee in the Faculty of Natural Sciences, thereby creating the enabling environment to train and develop students and junior staff to be future researchers. Prof. Niba specializes in entomological, pest and entomofaunal diversity and microinvertebrate survey work in the face of increasing adverse impacts of global climate change. Areas of special interest include the effects of landscape fragmentation on the distribution and diversity of insects and other invertebrates, as well as various aspects of environmental ecology with emphasis on using aquatic microinvertebrates as biological indicators for assessing the ecological integrity of freshwater systems. Research involving but not restricted to arthropod pest damage diagnostics, bionomical studies, specimen rearing identification, and invertebrate taxonomic studies is being undertaken under his supervision. His ability to collaborate with and attract research funding through the DST and NRF Center of Excellence for Invasive Biology, NRF Knowledge Interchange and Collaboration, Water Research Commission, the Walter Sisulu University Institutional Research Grant, and the NRF Tutuka Research Capacity Development Grant over a sustained period since 2008 has proven his capability to effectively source and manage such funding for personal and/or student research supervision until his rating as an NRF C3 rated in 2021. He has presented his research findings at various conferences and platforms locally and internationally and improved his research output as an established researcher. He is a member of the Entomological Society of Southern Africa and a former core team member of the DST NRF Center of Excellence for Invasive Biology from 2008 to 2012. He has reviewed articles for several accredited journals, including the American Journal of Experimental Agriculture, African Journal of Ecology, Journal of Applied Ecology, African Entomology, and external examined MSc PhD students from various universities in South Africa. The NRF has also invited him to participate in the institutional review of the NRF Research Cluster, 12th and 13th May 2015, Pretoria, and review grant applications for the NRF. Professor Niba has been the head of the Department of Biological and Environmental Sciences since 2021. I therefore, Prof. Nganywa, give to you our Honorable Professor, Professor Niba.
Thank you, Prof. Sorry, colleagues. Sorry, thank you very much for clapping hands without me saying clap hands, because now you're going to clap hands the way I want, which is not your way of. Okay? We're going to clap hands in a very special way here, because we're going to say, clouds are forming, rain is coming, thunderstorm. Let's do it. Clouds are forming, rain is coming, thunderstorm. That's a big thank you, Prof. And in those clapping of hands, we are also welcoming a professor, a full professor. Colleagues, that is not a small thing. A full professor. You know? Maybe to inspire the other people. Wait, Prof, I did not say stand up, please. Don't be in a hurry, because I'm gonna make you stand up in a special way, not in jail. Okay? I just want to explain that, you know, you earn professorship and you move up a ladder. You start by being a lecturer. You go and become a senior lecturer. You go and become an associate professor, which is very interesting because people call you professor without saying you are associate. But there is a time to grow to the fullest now. You become a full professor. So, universities, including our university is doing good things, colleagues, because not all universities are doing these inaugural lectures. This is a very good appreciation to our Vice Chancellor and the Council and everybody in management to think about this event. So once you are a full professor now, remember you are not writing any exams now. You are not going to be assessed now. So here we are assessing you. We are checking what are you made up of? Who are you? What are you specializing on? What makes you full? So we are celebrating you here. You are also celebrating yourself here. We are also listening. We are really very interesting to hear who you are. Why are you full? Are you really full? So please, Prof, I'm going to call you to come and share with us your food. Because we are really interested to hear what makes you full. In a very interesting way, Prof. Because I know people might not be interested in insects. Because insects, they bite us. But we are very much interested to know that what can happen. Where is the sustainability? Where are the issues? Where is the impact? We want to hear from your research. So we're going to call you. Music man, there at the back, do you have something that you're gonna make Prof check this, although the slides are checked or anything? You don't have, okay, it's fine. I'm gonna welcome you, Prof, to come and share with us. I think Prof wants to get a lecture from me now. I'm gonna sit down, Prof, now and talk less now. I'm going down, we are going up and come to the stage. Thank you. Let's welcome Prof and stand up, please, colleagues, please. I'm going to sit there now because I also want to listen.
Yes.
Thank you.
Thank you.
Over to you, Prof.
Thank you, Prof. Thank you. Thank you very much. Thank you very much. Yeah, the Vice Chancellor here be replaced by Professor Margaret Linington, Deputy Vice Chancellor Prof. Nosisi Feza, the Executive Dean of the Faculty of Natural Science, Prof. Modi, the Head of Department for Maths, Statistics and Computing, Dr. Panika Reka, the HOD for Chemical and Physical Sciences, Prof. Seki, and the Head and the HOD for Applied Sciences, Prof. Taziwa. And my wife here, beloved wife, Becky Nda Epus Niba, welcome. Distinguished ladies and gentlemen, I sincerely thank Walter Sisulu University for giving me this opportunity to deliver an inaugural lecture on behalf of the Faculty of Natural Science. I thank Prof. Modi for allowing me to deliver this lecture. I inculcated interest in entomology while in the Department of Biological Sciences, University of Calabar, Nigeria, under the mentorship of Professor J.C. Uswa, where I obtained a BSc honors in zoology, specializing in entomology. Thereafter, I was admitted to pursue a master's in agricultural entomology at the Department of Agricultural Biology, now crop protection and environmental studies, University of Ibadan, Nigeria, under the supervision of Professor Kola Ewete. Thereafter, I obtained a master's and PhD degree at the University of Natal and KwaZulu-Natal, respectively, under the supervision of Prof. Michael J. Samways. I'll be speaking to you this afternoon on the title, Navigating a Landscape of Applied Entomology and Invertebrate Conservation Ecology. What is entomology? Entomology is a branch of science that studies insects, whether in the pure or applied form, and more than 75% of all known living animal species are arthropods, that's invertebrates, with 95% of them being insects. What are insects? Virtually everyone in this audience lives with one form of insect or another, be it flies, mosquitoes, cockroaches, ants, butterflies, etc., at home, in the office, or in a restaurant. Therefore, we can all recognize a living creature known as insect. Technically, insects are invertebrates, animals without a backbone, belonging to the class Insecta in the phylum Arthropoda, the largest phylum in numerical size in the animal kingdom, accounting for over 75% of living things on Earth. Insect systematics. For any organism to be grouped into an insect, the body must be divided into three distinct parts: the head, the thorax, and the abdomen. The head bears a pair of compound eyes for sight and filaments, antennae for feeling the environment. The thorax is three-segmented, namely the prothorax, mesothorax, and metathorax, with legs attached to the thoracic segment. Consequently, insects possess 6 legs and are referred to as hexapoda. Biological success of these organisms. Madam Vice Chancellor, I know no other animal that possesses 6 legs. The closest relatives, ticks and mites, have 8 legs, 4 legs in toads, lizards, pets, primates, and ungulates. Insects are the most biologically successful and diverse animals in the animal kingdom. For instance, their small size accounts for their extremely low food requirement and colonization of small niches in their thousands. In addition, they are highly adaptive to extreme living conditions of hotness and dryness, ranging from the equator to the Arctic and subarctic regions of the world, being poikilothermic, cold-blooded. Besides, in their reproductive aspect, besides insects are highly fecund, producing many offsprings, and exhibit different types of reproduction, namely oviparous, ovoviviparous, viviparous, polyembryony, pedogenesis, parthenogenesis, and hermaphroditism. Oviparous reproduction is where insects are laid and hatch into young ones. Many insects practice this form of reproduction. In ovoviviparous reproduction, insects are retained within the insect's body until they hatch as they are laid. This occurs in most aphids and some flies. Viviparous reproduction is one in which embryos develop in the body cavity. When fully mature, they are brought forth alive, as found in man and other mammals. A good example is tsetse flies, Glossina species, which transmits sleeping sickness in cattle and man. Polyembryony is one in which many offspring emerge from a single egg. This is particularly common amongst endoparasitic insects that develop within their host tissues and are hence regarded as natural enemies. Pedogenesis is a form of reproduction in which immature stages that larvae mature their ovaries very fast and begin egg laying before attaining the adult stage, as it occurs in small midges. Pedogenesis is a type of reproduction in which young ones develop from unfertilized eggs. Even within this type, there are 3 different forms based on the sex of the offspring. It is teletoky if these offspring are exclusive female, arenotoxy if exclusive male, and amphiboly, both male and female offspring are produced. Hermaphroditism is the reproduction in which the insects produce spermatozoa and eggs, fertilized by the spermatozoa and develop into offspring. In contrast, the unfertilized eggs develop into males. Migration. Madam Vice Chancellor, I'm not attempting to bog down the audience with entomological jargon or make everyone an entomologist within this lecture. Still, I'm only exposing the complexity of these insect organisms. They are also the only known lower organisms with wings to conquer aerodynamic challenges by flying safely. In times of danger for any other exploratory activities to enhance their living. This is particularly true of the monarch butterfly, Dano species in Canada, which flies towards the south in late summer, where they mate, deposit eggs, and die. The newly emerged adults, the Stinera, fly upwards to Canada in spring. Role of insects to mankind. Madam Vice Chancellor, having highlighted what insects are and that they are the most successful animal groups on Earth, one can now proceed with the role of insects to mankind. This would help us discern those beneficial and harmful to man and agricultural conservation objectives. These insects contribute positively to human welfare and our well-being in several ways through their activities in the ecosystem. Pollinators. These are groups of insects that visit flowers of cultivated crops and other plants to collect nectar and pollen grains for food and manufacture other useful commodities for man's use. Notably among these are the honeybee as well as the butterflies, wasps, moths, flies, and beetles. In the process of visit, the process of the visit, they inadvertently transfer pollen grains to the stigma of the flowers and hence affect pollination. This has resulted in fruit and seed production on an unimaginable scale, leading to increased agricultural productivity and satisfying the food needs of man. Manufacturers of small commodities. Different insect species across various orders are manufacturing several useful products. This includes the following: honey, propolis, and beeswax. These are products manufactured by the honeybee Apis mellifera. Honey is helpful as a food source and for the medicinal treatment of wounds or burns by preventing bacterial infection. The beeswax from hives is used in the production of candles. Beekeeping is a big business worldwide. And wax, this is a commodity from the nests of wasps and used in the manufacture of candles and polish. Shellac, this is a commodity obtained from scale insects, Hemiptera cycadi, and used in the making of paints as well as gums for sticking motor engine gaskets together. And last, cocoon. This is a woven silken material produced by the larvae of Lepidoptera insects, such as Anaphis venta, when about to pupate. The harvested cocoons are unwound into yarns and used in sericulture for making cloth, silk cloth, and traditional dresses. This is another commercial enterprise in which individuals and corporate bodies can generate revenue. Natural enemies. These are groups of insects that kill other insects which infest our agricultural products. They are the predators and parasites that feed on harmful insects on our crops. These predators are generally bigger than the prey, while parasites are smaller than their hosts. Others are ladybird beetles, Chilomys lunata, lacewings, Neuroptera, and dragonflies, Odonata. In the coccinellid beetle, ladybird, assassin bugs, and mantis, both adults and larvae or nymphs, kill and feed on prey, while only larvae are involved in Neuroptera and Odonata. Biocontrol. The parasitic insects, especially the endoparasites, are found in two insect orders, Diptera and Hymenoptera. These include the bracconid wasps as their slender ovipositor to deposit an egg within the tissue of their host, later hatching into a young larvae, which continue to feed within the host tissues and eventually burst out the host tissue to pupate outside, with the adult later emerging. In this way, the host is killed. It therefore follows that this group of entomophagous insects that help in reducing insect pest population to sub-economic levels can be manipulated for the control of major insect pests through a control strategy called biological control. These are examples of your bracconid wasps. Use of biocontrol. And soil nutrient enrichment. Certain soil-dwelling or subterranean insects such as springtails, collembola, as well as dung beetles enrich the soil by degrading plant tissue and or mixing feces with the soil to increase the soil organic matter content, which enhances crop growth and subsequently yield increases. This is of great advantage to farmers who most often practice zero tillage. The larvae of some lepidopterans, butterflies and moths, as well as larvae and adults of the beetles, palm weevils, palm beetles, the winged termites and queen termites, adult crickets, some grasshoppers, and locusts are used as food to man. It's an example of a larva of the beetle as we use source of food to man. Insects in aesthetics. Insects are very beautiful. The combination of color displayed by butterflies, moths, and other insects such as beetles, dragonflies, and grasshoppers depicts the work of nature. A mounted insect display in the home adds color and serenity to the immediate environment. There's an insect species given the scientific name Cynthia species. Its common name being painted lady, And this is without prejudice to ladies who bear Cynthia in this audience. Madam Vice Chancellor, you know that ladies can be beautiful when they wear makeups but are not painted. Artists are also use insects as designs in the textile industry, and they are also used natural enemies would allow them to multiply unchecked until they attain pest status. This is clear in the citrus scale insect Achaea pachasi, which was exported along with citrus varieties from Australia to California in the USA, leaving behind its natural enemy. Rhodelia cardinalis. It was not until this natural enemy was exported to California to deal with the scale insect that it was eradicated. This was the first case of biological control of insects. The first classical case of biological control of insects. Insects related to crop commodities and plant health. It is convenient to group crops according to their commodities they supply and for pest identification and management purposes. Thus, there are groups such as cereal, legumes, vegetables, pomology, floriculture, tree crops, and store products. Insect pests infest each group to varying degrees, some as major pests and others as minor pests, all impairing the growth and productivity of such crops, culminating in yield and quality reduction. Research work I have undertaken on some major insects attacking crops will suffice as my contribution to knowledge in this domain. Cereal stem borer assembly structure in a subtropical ecosystem, Eastern Cape. The most important cereal crops cultivated in Africa include maize, millet, guinea corn, and wheat. They are good carbohydrate sources in human diets and animals' feeds and supply the needed energy. What is the importance of elevation in this study? Elevation is one of the several factors influencing insect communities generally. Moreover, particular microhabitat conditions may favor the coexistence of particular groups as different elevational bands. Climate affects species distribution through primary productivity, which correlates with moisture and temperature. Species at higher elevation gradients are tolerant to a broader range of climatic conditions, such as more significant temporal variability in humidity, precipitation, and temperature than at lower elevations. Insect population can also be affected by elevation directly through its influence on biological interactions. For example, some natural enemies such as parasitic wasps are abundant at certain elevation bands. This could be due to the displacement of populations in response to climate change, resulting in a mismatch between predators and prey and between parasitoids and their insect hosts. Studies have shown that species ranges decrease with increasing elevation, although McCain and Knight suggest that this is not the case for some plants and vertebrates. Since non-migratory individuals must be physiological or behavioral capable of tolerating a full range of environmental and seasonal changes, natural selection therefore favors tolerant species at higher altitudes. High temperatures may also lower survival of species temperature threshold. High temperatures may also lower survival of the species if temperature threshold is exceeded. Early warning systems. Vulnerability assessments conducted by the Early Warning System Network in August 2013 indicated that 7.7 million people in South Africa developed community, in the South African Development Community, SADC region, are at risk of food insecurity. The Institute of Risk Management proclaimed that prolonged strikes, fluctuations in crude oil prices, load shedding, lack of skill and innovation, and immigration of highly skilled personnel increasingly affect the South African economy adversely. The IRM also found that workers lost enormous wages due to strike actions. High food prices and dwindling resource of disposable income for South African households would exacerbate this situation if nothing is done to boost the agricultural sector of the economy, including securing basic food crops such as cereals. This situation may worsen. reduction in cereal production due to stem borers. Insect pests, especially Lepidoptera stem borers, significantly reduce yield, quality, and quantity in cereal production locally and globally. So far, few studies have been undertaken to deal with maize yield losses attributed to these pests in subsistence farming communities in South Africa, which have no purchasing power to buy genetically modified organisms, stem borer resistant maize varieties, particularly in the Eastern Cape province. Baseline data and information about economically important insect pests are critical for establishing long-term integrated pest management programs. And this section of the study aimed to identify Lepidoptera stem borer species associated with cultivated grain crops and wild animals and wild alternative host plants at 3 elevation gradients in the former Transkei area of the Eastern Cape of South Africa. Stem borer sampling was undertaken during the maize crop and non-crop cropping seasons in Port St. John's, Mthatha, and Elliot, which differ in elevation above sea level, annual precipitation, and temperature. Morphologically identified borers retrieved from each sampling unit were recorded, processed, and read in lab to adult stage for identification at ambient temperature range of 250 to 280 degrees centigrade and a relative humidity range of 65 to 80%. Specimens were sorted to morphospecies, M1 to M6, MS1 to MS6, based on external morphological characteristics. 6 stem borer species, Brucella fusca, Sesamia calamistis, Eldana saccharina, Chilo oricacolis, Chilo saprophagus, and Chilo patellus were collected during the survey. Eldana saccharina and Chilo oricacolis were absent in Port St. John's, while E. saccharina occurred in very small numbers in Elliot, where C. patellus was absent. Brucella fusca was more abundant at high altitudes. It's in Elliot. It's a picture of the abundance was actually very high, was highest in Mthatha, and followed by Port St. John's and Elliot. Borer community structure in the study showed a similarity between Mthatha and Elliot. Species richness abundance across study sites was not statistically significant. And when designing control strategy for borer control, elevation may not be a major concern at this spatial scale. Nutritional plant protein from cowpea, Vigna unguiculata. Cowpea is the 3rd most important grain legume in South Africa after groundnuts and dry beans. It contributes immensely to the protein diet of most poor families, provides income to farmers for the sale of grain and fodder, and is a good source of animal fodder. Although cultivated primarily for edible seeds, direct consumption of cowpea leaves and seeds in different parts of Africa provides nutritional plant protein, especially in some areas of the former Transkei area of South Africa. The phenology of cowpea comprises 4 main stages: pre-flowering, vegetative, flowering, pod formation, and pod maturity. Insect pests pose the greatest threat to cowpea cultivation and attack the crop from seedling germination to storage. Natural enemies play an important role in limiting potential pest population surges. However, in a cowpea monocrop system, there are not usually sufficient numbers to suppress high pest population infestations. Exploratory control using natural enemies. Although pest biology on cowpea had been studied extensively, agroclimatic conditions differ completely amongst agroecosystems, and these differences could influence the population fluctuation of insect pests. A limited amount of work has been done to understand the pest biology of cowpea in South Africa, especially the former Transkei area. Therefore, a yield investigation to test 5 varieties of cowpea with different phenologies was undertaken to determine when and which insect pests occur on the varieties, and to take an inventory of the natural enemies with the potential to suppress the pests. These are actually characteristics of varieties used. And there are 5 varieties. They're Glenda, Betrona Whites. They are all local varieties. Ife Brown is a variety from Nigeria. And the ITO33K 3693 and ITOK 4K 221 are from ITA in Nigeria. And they show variation in maturity and growth habits. Some are erect, some are runners. And 3 varieties show photosensitivity and 2 show non-photosensitivity. The phenologies of the cowpea varieties. In fact, we'll find that there were statistically significant differences between the 5 varieties with respect to days to 50% flowering, days to 50% ripening, and days to 50% maturity at different probability levels of 0.1, 0.5, and 0.001. Low insect populations as vegetative suggests that few insects specialize in leaf consumption. In contrast, high population counts at low flowering and pod formation suggest that more insect species have a predilection for these 2 phenological stages. Low population counts at pod maturity may result from reduced food resource availability and sporadic spray regimes, amongst other factors. The peak population at flowering may be attributed to the high abundance of Anthophilus coleoptera species that prefer inflorescences, flowers and flower buds as feeding sites where they get high nutrient quality. And the taxonomic profile of insects on the crops, the group Lepidoptera and Homoptera. Lepidoptera, the family Pyralidae, the species Maruca tesulalis and Aphis craxivora occurred at the seedling and pod stages. And the Coleoptera, Myridi, and Melody, they occurred mostly at flower and pod stage. And the Hemiptera, Alididae, Pentatomidi, and Corydii were restricted to pod stage. Species were Mepirus jaculus, Riptorchus dentipis, Aspavia species, Nezara viridula, and Clavilla tomentosicolis. They were mostly recorded during the flowering stages. The data on weekly mean density of aphids, Lepidopteran larvae, blister beetles, and pod-sucking bugs show that aphids and Lepidopteran larvae were the first major pests to enter the cowpea crop during the early vegetative stage. Aphids colonized swiftly and attained the maximum population within 3 weeks of the colonization of the cowpea field. It can be inferred that the softness, succulence of the plant tissues during early vegetative growth may have attracted these sucking pests. Green caterpillars feed on tender foliage at early vegetative stage of the cowpea. This just shows a general overview of the infestation levels from three sites, Water Slough, University Solo, and Efata. You can see they are actually similar, attaining peak infestation between 6 and 13 weeks. And we see the aphids, followed by the Lepidopterans and the pod-sucking bugs. They are actually between 6 and 13 weeks that we get peak infestations. And what are the implications for pest control? Amongst the arthropod natural enemies recorded in cowpea fields, this coccinellate Chilomys lunata had been reported as a predator of aphids in South Africa. However, it is not solely effective in protecting the susceptible varieties from damage. The assassin bugs and Rhinocoris segmentarius are generally predators on legumes and rice throughout Africa. The results presented here indicate that the activity of most cowpea pests is concentrated between the 8th and the 13 weeks after sowing to coincide with flower flower budding and pod formation stages when there is sufficient food resource availability. If arthropod natural enemies are not present in the crop in sufficient numbers to control increasing pest population, it would be better to use other control measures targeting the flowering and the pod formation stages. Madam Vice Chancellor, permit me to indicate here that insects are invertebrates that contribute to over 75% of biological control on Earth. South Africa is one of the world's most mega-diverse countries, meaning it is especially rich in biodiversity. This wealth of information underpins a large proportion of the economy, and many urban and rural people are directly dependent on it for their livelihoods, jobs, food, shelter, medicines, and spiritual well-being. Sustainable use and management of South Africa's biodiversity requires a solid knowledge base and access to relevant information and data. Work done so far. Researchers in South Africa have made considerable progress towards documenting biodiversity. However, large gaps in our knowledge still exist, and it has been estimated that more than 50,000 species remain undiscovered or undescribed. These species may have economic benefits, or they could play a critical role in the functioning of ecosystems. In several parts of South Africa, biodiversity has only been sufficiently explored. Biodiversity has only been superficially explored, so data critical for development planning and ecosystem management are poor. The distribution and abundance of most species, including invertebrates, in South Africa are also far from complete, even for the better-known groups, including plants, small mammals, reptiles, and amphibians, which make understanding change in status and use problematic. For the knowledge that has been and continues to be generated, there is generally poor coordination and is not all readily available to stakeholders who currently or potentially need it. A new long-term program has been developed to address these challenges. The program's primary focus is to generate, coordinate, and make accessible knowledge relevant to essential biodiversity variables, including occurrence, species diversity, population abundance, phylogenetic information, and barcoding. This type of information is often referred to as fundamental or foundational because it forms the basis of many other aspects of biodiversity research and decision making. These data sets are critical for ecosystem mapping, monitoring, and reporting on the state of biodiversity, for sustainable use of biodiversity, and understanding and mitigating the impacts of global change on biodiversity. Environmental scan. The program will deliver products that contribute to the fulfillment of the objectives included in the key targets of the strategy of the Convention on Biological Diversity, the Global Taxonomy Initiative, or Convention on Biodiversity, the National Biodiversity Act, the National Biodiversity Strategy and Action Plan, the Global Change and Bioeconomic Grand Challenge, and DSI, and its program on indigenous knowledge systems. The program's output is fundamental to protecting South Africa's economic infrastructure, on which many industries and communities depend for their livelihoods, and supporting the sustainable use of components of biodiversity. Several workshops, some of which I attended and participated, involve DST, NRF SANBI, and representatives of the program such as SANBI, ECOL, and South African International Barcode of Life, were held in 2011 and 2012 to discuss the DST request for an integrated program which would reduce transaction costs of separate programs and increase impact. This group formed a task team which developed the program concept. In 2012, I attended a brainstorming session for the users of foundational biodiversity information in order to identify priority needs for the program or decision making. Focus areas. The program had four main strategic objectives which deal with the generation of information, the mobilization of information, the integration of data, ensuring the management and dissemination of information, capacity development, and the development of an understanding of how best to ensure the update and application of outputs in foundational biodiversity knowledge. Research Objective 3 to attract, develop, and upskill people to ensure appropriate capacity for developing for biodiversity information generation, dissemination, and application is quite interesting. This includes training of postgraduate students in the generation, management, dissemination, and application of foundational biodiversity knowledge, development of capacity for application and use of knowledge or data amongst practitioners, provision of opportunities for emerging researchers, training of research data managers who work with foundational biodiversity information in capturing and managing of data, and training of researchers in novel approaches to identify biological material. My research and that of the postgrad students I developed had been focused on documenting the richness and diversity of soil surface dwelling invertebrate fauna within protected areas, that's natural parks and natural reserves, freshwater lentic systems of South Africa. Determine what variables or intrinsic species factors account for species absences and presence, and assessing how these systems can be better managed to protect species in their habitats and their habitats over time. Species lists and recommendations from the research are provided to research managers and conservation practitioners and planners. Multi-taxa approach is Seleka Natural Reserve. Using a multi-taxa approach, invertebrate species of Seleka Natural Reserves were sampled under the following theoretical framework background. Species richness increases with increasing microhabitat complexity for various fauna groups. Any reduction in habitat complexity and niche availability may thus induce strong competitive interaction between fauna species, resulting in their extinction. Additionally, abiotic features of natural environment, such as soil chemical constituents, are influenced in different ways by plant species. In the process of high cation uptake and releasing organic acids and oxidates, plants can change local soil pH. Plant biomass over the leaf cover and leaf litter deposition may influence local soil and ground surface temperatures, such as the susceptibility of the soil to erosion, effectively providing a buffer against the infiltration of solar radiation and wind. Differences in the vegetation form of natural communities can greatly alter various components of the abiotic environment relative to natural conditions. The case for terrestrial invertebrates. This in turn may affect the biodiversity and trophic interactions within fauna communities, where environmental parameters may shift away from optimal levels essential for food survival, for species survival. South Africa has been ranked the third most biologically diverse country in the world, based on an index of species richness and endemicity. Still, only 6% of the country's biodiversity is formally conserved. Outside of these conservation areas, many habitats are threatened by urbanization, industrialization, agriculture, pollution, and overharvesting. Although the country's invertebrate species richness is essential, it is estimated at twice to 3 times the present described number of 70,000 species, there is still a vast gap in the understanding of what the invertebrate component of biodiversity consists of, where it occurs, which species are threatened, and where they are. In addition, insects are largely ignored in ecotourism industry, yet many of the larger, more spectacular, interesting, and rare invertebrates have potential for incorporation into community ecotourism activities. Little is known about the composition and distribution of invertebrate fauna groups within protected areas of South Africa. The Eastern Cape Province falls within the Albany Center of Endemism and contains all 7 biomes of South Africa that potentially provide ideal habitats for a diverse group of invertebrates. Seleka Natural Reserve is one of 13 protected areas in the province aimed at protecting local biodiversity. The reserve was preferred for this study because of its distinctly varied and heterogeneous habitat conditions, potentially attracting a rich diversity of invertebrate assemblages in one location. Furthermore, Seleka Natural Reserve is situated at sea level, enabling it to attract fauna elements predominantly from higher and lower elevation gradients. The study aimed to identify the composition and distribution patterns of the invertebrate assemblage within the reserve, determine environmental variables that may or may not influence species distribution across distribution patterns, and based on the findings of the study, propose management recommendations for conserving the invertebrate fauna of the reserve. Madam Vice Chancellor, distribution in habitat complexity and niche availability affect competitive interaction between fauna species, which may result in their extinction. This study was conducted to assess the conservation value of 4 a priori selected sites. Site A was minimally disturbed natural forest patch, Site B, herbaceous grassland patch, Site C, road verge patch, and Site D, highly disturbed natural habitat patch using epidemic invertebrates in Sri Lanka Nature Reserve. And a total of 2,415 species belonging to the phyla Arthropoda, Mollusca, and Annelida were caught from all sites using pitfall traps. These were sorted into 14 orders, 15 families, 21 species, and 17 morphospecies. This table shows the number of surface-dwelling invertebrates, the Formicidae, Carabidae, Cuculinidae, Cerambicidae, Staphylinidea. They were actually insects, as opposed to the rest of the ones down there on the list, like your Zodaridae, Thomisidae, Scottidae, Sortisidae, and so on, that were non-insect taxa. And we'll notice that those ones are actually heavy infestations. And this actually shows environmental variables and the canonical corresponding analysis axis for terrestrial invertebrate assemblage across Sri Lanka Nature Reserve. Axis 1, those variables intersect correlations. Axis 1, litter depth, soil phosphorus, soil pH. They were extracted as very important environmental variables that could be relevant in the distribution of the species that were sampled. Discussion and conservation implications. Multivariate approaches using canonical corresponding analysis show that some measure site variables, litter depth, grazing intensity, soil pH, soil phosphorus, and soil texture accounted for species distribution trends across the sites. Site A had common widespread species, widespread and rare typical invertebrate taxa, and therefore was of high value for conserving invertebrate biotic communities within the reserve. The other 3 sites also offered optimal and unique habitat requirements for protecting and preserving species such as Ancylotrypha zurensi, Lycosidae species 2, and Opilionis. It was recommended that biotope management in the reserve should therefore aim at preserving a variety of habitat patches that differ in structural and compositional vegetation. Furthermore, anthropogenic pressures resulting from frequent clearing should be regulated, especially during summer, to cater for various developmental stages of the invertebrate assemblage of the reserve. Biomonitoring of the Umtata River. Madam Vice Chancellor, many South African rivers, including the Umtata River, have deteriorated in quality in the past few decades, mainly due to increased industrial, agricultural, and domestic activities within the catchments. Such activities alter its physiochemical characteristics and ecological integrity of the river. Since different benthic taxa tend to exhibit different tolerance levels to individual environmental parameters, water of suitable quality is essential to maintain healthy populations of benthic fauna. In the upper section, with extensive pine plantations in the headwaters and clay loam section of Langeni, and lower down the river. The study assessed the Umtata River's ecological state and impact of water quality on benthic macroinvertebrate communities from 2015 to 2016. Fauna species specimens were sampled and environmental variables measured at 4 sites, including headwaters, Langeni, Kanbi, and Silaka, that vary in topography and human impact along the river. 2,980 specimens were recorded as sites and classified into 12 orders, 46 families, and 71 species. Species composition and distribution were directly or indirectly affected by season, physiochemical variables, and habitat heterogeneity. CCA results show that electrical conductivity, pH, temperature, soil, dissolved oxygen, and vegetation accounted for species distribution patterns. Both habitat-restricted and common widespread species can be used as indicators for assessing the quality of the Mtata River system. Biomonitoring implications of the study. Habitat patch level management along the Mtata River should aim at preserving sufficient heterogeneous indigenous vegetation categories, riparian, aquatic, and algae, especially during the summer season when habitat requirements are optimal for most growth stages of the benthic macroinvertebrate taxa. Higher family and species richness and abundance for protein-sensitive members of the Ephemeroptera, Trichoptera, and Odonata group were mainly recorded at headwaters and Langeni sample units. The habitat at Kambi and Nsaka, lower down the river, needs to be improved to approach natural conditions. Conservation implications. Both rare, habitat-restricted, and common widespread benthic macroinvertebrates identified in this study can be used as indicators for assessing the ecological integrity of the Mtata River system. The practicality of using species-level surveys on a routine basis can be challenging due to a lack of available taxonomic expertise and time constraints. It may be advisable to select a few indicator species identified in this study and to monitor them as surrogates for the whole community. Conclusion. Research into the genetic diversity of cereal stem borers needs to be expanded to include complementing such studies with conventional morphological taxonomic studies in identifying both species that are new, borer species that are new to science. New improved varieties as high-yielding cowpea varieties are adapted to the South African agro-ecosystem must be tested and compared with novel varieties, and research on adaptive high-yielding species should be focused on to alleviate poverty and hunger. In the face of increasing adverse effects of global change on local and global biodiversity, it is essential that there is a continuous investigation on species richness and diversity trends of species and their habitats on the ecology of terrestrial and freshwater systems for sustainable conservation action. So thank you very much.
Colleagues, can we clap in our fashionable way? Clouds are coming, clouds are coming, rain is coming, rain is coming. Thunderstorm! Thank you very much, Prof.
Thank you.
I won't allow you to sit down yet. I will ask at this moment, Dr. Tapira, where is the trophy? Oh, okay. Yes, I see the respondent, but I want, before Prof can get the respondent and sit down, I want to change the program a bit and have Prof Lymington to come and give the trophy to Prof, so that Prof can sit down. Yes.
Thank you.
So you can come here on stage together.
I like the idea of a trophy, that's nice.
That's great. Thank you very much. Oh, wow. Thank you very much, Prof, for the speech. Thank you very much. Now we're going to move to our academic respondent. Colleagues, now for this program, you can get seated. Thank you, sir. For this program, the longest speech is the speech of the inaugural lecture. All the other speeches behind that one are sharp, ne? Sharp, so that it does not dominate the original speech. But I trust the other speakers after Prof that they are going to it's not their day. It's your day, Prof. So that's why we are not disturbing you. But if a speaker is speaking long, I will stand up there so that I say, it's enough. But I want the people to be very brief now. Thank you very much. I'm going to call our academic respondent, Professor Simon Kamande Kuria, an Associate Professor of Entomology, to come and give us key things about this speech.
Okay, thank you. Madam Vice Chancellor, Madam Deputy Vice Chancellor, University Registrar, the Director, Research and Innovation, Executive Deans and Deputy Executive Deans present, HODs, colleagues, dear students, ladies and gentlemen, good afternoon. Mwaloeni. It gives me great pleasure to respond to Professor Niba, Professorial Inaugural Lecture, entitled Navigating the Landscape of Applied Entomology and Invertebrate Conservation Ecology. Let me assure you from the first instance that it will not take long. I will try to avoid the jargons so that we are together. And in short, I'll try to kind of address those among us who are not entomologists, and therefore they can appreciate what Professor Niba has gone through. In short, for me, Professor Niba started from the basics of entomology and eventually to the applied entomology and to the application of entomology. In short, he told you what is entomology. I will not repeat it. He said it's a study of insects, so all of us know insects. And if you don't know insects, I think he mentioned some, I mentioned one or two or three. You must know the locust, I'm sure, isn't it? You must know the butterflies, I'm sure, isn't it? And you must know the cockroaches, etc. So among them, and that the insects, they cut across our lives, and almost all of us, in one way or the other, we are entomologists. You have dealt with the insects at one time or the other. Then he went ahead and possibly described about the reproduction of the insects. There are so many types of reproduction, I'll just give you two only. One, they saw the sexual and the asexual. The sexual is the one that we know, that is the male and the female, and the other one is where there may be no male, but the female is able to reproduce without a male. The only thing the interesting bit I want to tell you about the insects is this. Generally, is that the insects do not care of the young ones, apart from the social insects. If we are together. So the social insects take care of the young ones more than human beings. I'm telling you that the king and the king is only for mating, the queen is only for laying eggs. Then there is the workers and soldiers. Theirs is to protect the young ones and to feed and clean for the young ones and everything else. I hope we are together. So therefore, those young ones of the insects, you cannot get in touch with them. Together, then we go to the other one whereby the insects do not care of the young ones. However, they are getting better maybe than some of our human beings for that matter. That they don't take care of the young ones. They make sure where they lay their eggs. The eggs are, when they hatch, the young ones are able to get food. And therefore, once they get food and they grow, they can be able to go and look for their own food. I will get it. So then that's a bit about the reproduction, and then we went about and talked about the beneficial insects. I'm not sure which one I'll talk about, but let me talk of the very common one, the pollinators. All of us know about the pollinators, mainly the bees, the wasps, and the butterflies, etc., ants, beetles, etc. And all of us, they know the depends on the pollinators. And therefore, what we know, given the effect that we have, especially the anthropogenic activities on the environment, which affects the environment, if very soon or in the future we may have a problem if there are no insects, then we may have no crops, some of the crops producing because the pollination cannot take place because the insects assess or assist the pollination. In short, and I'm sure some of you know, at the moment, there are people who their function is only to rear bees, specifically for hire, because the people who are having vineyards, their vineyards cannot, there are no insects to pollinate them, so they go hire the bees. The bees are transported or transferred to that farm. They do the pollination, then the owner can take away the bees to go and get his honey in another place. So in short, we better conserve the insects for our own survival. I'll go to the other aspect of the importance of the beneficial insects of insects is the source of food, and especially for human food. I'm sure in this country all of you know about the Mpana worms. Yes. So in South Africa there are worms that people feed on, and therefore they are good source of food. And therefore, with the scarcity of food getting a problem and the proteins becoming costly, we may rely on insects to supplement the proteins. In other countries, for example, in East Africa, they feed on termites and locusts, and this is also in some other countries. And there is research currently that crickets are also very a delicacy. I'm done with the beneficial insects. The harmful insects, again, there are so many, but I don't want to deal with them. Let me deal one or two. The harmful one, and I'll deal with the sassaflies. The sassaflies, there are few in this country. I don't know whether they were eradicated or not. But the sassaflies are vectors of trypanosomes, which is a disease for both animals and human beings. And therefore, the insects have decided to cooperate with the pathogens to see how they can fight the human beings, and therefore, they are able to transmit the pests to us, the pathogens to us, the human beings and the animals. And the disease can be lethal, eventually meaning that you can die. Let me say I'm done with the harmful insects. I think you got a lot of it. And therefore, I narrow down to some of the research Professor Niba and his students have carried out, which he dealt a lot with, but I'll try to minimize my stories. He talked about the Lepidoptera stem borers. I'm sure some of you are wondering, what are these stem borers? These are the caterpillars, my friends. And these are the caterpillars. So these are the larvae of the moths. In a few cases, the larvae of beetles can do the same. And therefore, there are many, in fact, there are those that attack the sugarcane, there are those that attack lice, but Professor Niba and his students worked on the stem borers that attack the maize. And what they found out is between Port John's, Mtata, and Elliot, there were about 6 species, and more or less the same species were occurring in the same place. However, there was only a slight difference in one case that the Bucilla fusica, one of the species, was in higher land, that's in Elliot, as compared to the other areas. I think I'm done with that. I don't want to give you a lot on it. And then they have also worked on the cowpeas. I think you heard it. And what they were trying to do is this, that they were trying to find out whether the varieties which have been developed could be able maybe to give us more yield and be less susceptible to pests. Unfortunately, most of you know, the agronomists, they rarely remember about the pests. And what they do is you come up with a crop, it can give us very good yield. Unfortunately, it's also very preferred by the pests. And therefore, you want to grow the crop, but the pests cannot allow you to do it. And therefore, they found out, yes, these varieties were more or less the same. All of them more or less were being attacked by the pests, though some of them had good yield. And they realized, yes, the natural enemies could control the pests in one or the other, but they needed to be supplemented, especially between the pod formation and flowering. And therefore, with those, the natural enemies working, they need to be supplemented to control the pest. And more or less, and finally, I'm almost done, Prof. He has also done a lot of work in ecology and conservation. And what the work they've done is to look at what we call the biodiversity of invertebrates and also the insects found on protected areas, what is popularly known as the nature reserves. And the essence of this is to really understand because you cannot conserve what you don't know. What is there? What is these invertebrates that are in these particular nature reserves? And which characteristics and the characteristics that affect these invertebrates. That way, we are able to know what is there in those nature reserves. But then the point is, what's the importance of preserving these insects or these invertebrates? Very simple. As we destroyed and protected the environment, the only savior in future is these protected areas. The insects and other invertebrates will be able to move from those areas and maybe try to occupy the areas which we have destroyed, or in case in future we decide to rehabilitate them, we have a source of these insects, lest they get extinct. And finally, they did some work on freshwater, whereby again they looked at the invertebrates found in freshwater and how they can be affected by the different physical-chemical parameters. And for sure, they found that some of the insects were affected by the physical-chemical parameters. What does that mean? Is that water is likely to be polluted, and therefore you could use these particular insects as bioindicators to know that this water is polluted, and therefore maybe you may try to minimize the damage. With those many comments, thank you. Thank you.
Thank you very much, Prof. Colleagues, I like the interpretation from Prof because it made it so simple, what we learned from Prof. And also to add, if I was also a chosen respondent, I would look at this conclusion that I think it is deliberate to stay there. And from that conclusion, I see some of the keywords that I was looking for, Prof. Because I was looking for any sustainable development goal, and I could see alleviating poverty and hunger. I was happy there. And I also saw sustainable conservation action. And I also saw climate change, global climate change. I was so happy. Then those are the words I was talking about, Prof, on impact. Thank you very much for your presentation. Thank you very much, Prof, for that response. We really appreciate it. Now I am going to call the Executive Dean of the faculty. Don't stand up before I call you, Prof. The Executive Dean of the faculty to congratulate our colleague. Professor Modi is the Executive Dean of the Faculty of Natural Science. And now he is going to come up and take a stand and congratulate our Professor Neighbor for today. Over to you, Prof. You can stand up. Can we clap hands for Prof?
Thank you for the opportunity. I know that I'm not here to have a word of thanks, but I feel compelled to say thank you to the Vice Chancellor who permitted this day to happen. I feel compelled to say thank you to Professor Nganyo and her team, not only from the Directorate of Research, but also the Management and Communications Division. I feel compelled to say thank you to my line managers, Professor Lennington and Professor Feza, as well as colleagues who came to make this day the one that we want it to be like, visited by professors from outside the faculty and everyone. I'm not taking the role of the person who's going to do a vote of thanks, but I feel like to a certain extent I'm the host. Professor Niba, congratulations. It has already been said that the path to a full professorship is a long one. Not everyone finishes the journey as a full professor. Not everyone who starts the journey aims to finish it as a full professor. It happens, and no one works very hard to be a full professor through their contribution of knowledge. But it does happen. In some instances, we are told that we are full professors now. So thank you to whoever assisted you from the time you started as an undergraduate student, postgraduate student, your mentors, and I'm sure many people, but most importantly, the people that you trained after your PhD are actually the ones who took you to this level and the colleagues that you collaborated with. And lastly, thank you for articulating the strategy of the Faculty of Natural Sciences. You made it very clear that this faculty is relevant to every faculty at WSU. And this is something that I always say. I mean, you talked about economic injury level. You know, the finance people can hear that. You talked about the ho-ho that eats the ho-ho that eats my crop. Do you understand? So thank you very much, Prof. And I know you're going to continue to pursue the energy of assisting your students, working with your colleagues, but also to respond to the many questions that this province, this country, and this world requires answers for, particularly from your area of study. Thank you very much. Congratulations. Let's shake hands.
We are enjoying it. I also want to take it. I think I must also do this. Yes. You must carry this all the time. If it's sleeping. Can you achieve that?
Let's call it.
That's nice. Thank you.
Yes, exactly. I don't have something like this. I would never have it.
Who wants to shake hands with me? Take a peek. This is important. Thank you very much. You know, when I graduated my PhD, they told me that I'm wearing a gown. So I slept with that gown because it was a gown. So please sleep with that one tonight. Colleagues, I'm going to call Professor Lennington again to come and do a vote of thanks. Before that, Prof, I just have a special thank for myself, to the audience and everybody, but I want to pick up someone special, Mrs. Niba, because, you know, colleagues, let me share something about your partner. You know, in this journey of ours as academics, the most person that feels the pain of you not being there is your partner. Because every time you go away, every time you are concentrating on your books, every time you are doing something that is making you far in your research, this person is suffering. So that suffering pays out today. Can you see Can you see, my love? So I'm very thankful to you for the support you gave to your husband, because, colleagues, we really need the support of our partners to be in this journey. It is a very difficult journey, but your partner can take you far. So please, it's my special thanks, Prof, to the partner. Yeah, so I'm going to call my DVC, Prof. Lidington, to come and do a vote of thanks to all of us. Over to you, Prof.
Thank you, Program Director. I think I must start with saying, Prof. Modi, I feel compelled to congratulate Professor Niba. Since everyone's been compelled to do my job, I'll do yours. I want to start off by thanking Professor Niba for his really interesting inaugural address. I think you let the audience into insects so that they could actually understand the importance of insects and how they're so fundamental to life. And I've got a passion for biodiversity, and I always say, if anything nature teaches us is that diversity is important. Without it, we die. And we must celebrate diversity. So thank you for your inaugural address. I think was very much a celebration of insects, both the good and the bad, and how little we know and how many there are. And I just thought it was interesting that there are 10 quillion insects on Earth. That quillion's got 19 zeros. It's a 1 with 19 zeros. So it's 20. It's not a 6-figure number. It's a 20-figure number, 20-digit number. That means that for every person on Earth, if you're trying to figure out what a quillion is, or 10 quillion, there is 1.2 billion insects per person on Earth. So if we think we're important, I think they're more important. There's much more than us. But they are important with their biodiversity, and I think you've explained it really nice, the issue with the rivers, the climate change, and that number gives us our adaptability. Should things change, we have enough insects to change with it, because without it, we lose all those benefits which you put so nicely together. I also want to thank the respondent who also gave us an insight to how important this type of research is, and why we are actually privileged to be able to listen today. And thank you for that perspective, and for it's always good to hear from the person who isn't presenting the research that the research is important. So thank you very much for that. And I think as a last comment, because from what Prof. Modi was saying as well, there's I can't remember who it was, some American, I think it was Carnegie, but I could be wrong. He said agriculture is the beginning of all culture. So until man learned to till the soil, he had no time for music and songs and poetry. So agriculture is the basis of all academia. He also says some other things like, but I won't repeat them because the economists hate it, because he says agriculture is the only true discipline, all the others are frauds. But anyway, but we won't go there. I want to thank as well, after Prof. Nganywa has done so, Mrs. Niba, thank you for allowing him to live his passion. People become full professors because they're passionate about that. And I think in allowing him to do it, you also, I know it's a lot of sacrifice, but you also receive a lot of joy, because he's a happy person following his dreams. So thank you for your support, and to your family. I I would like to thank everyone here today. Professor apologizes. She has a flight because she's doing a presentation in Joburg tomorrow, and the last flight out was 6, and she couldn't miss that because then she misses her presentation because the first flight isn't early enough. So she apologizes, but I want to thank her for being here and her directorate as well for being the organizers together with MCD for today, and all the people who did the hard work, the photographers, the sound engineers, and all those people in the background who have been here today, who are so important. I'd like to thank Prof. Modi, all the HODs, the faculty present, as well as the students present. And I'd like to thank the two professors from humanities. Thank you for being here. Not your field, but I hope you learned a lot. It was really very inspiring. So that's, so I don't think I have anyone else to thank. I hope not. If I've forgotten anyone else, I apologize, and you're noted and thanked. The joy that Prof. Niebuhr expressed when he gave his lecture, I think, shows his thanks to everyone who's been involved in his journey from beginning to end. So thank you very much.
So, as the program director, let me thank you, Professor Lennington, because you gave this day a dignity it deserved. So I am so happy about what you did by coming and also executing everything you did today in success. I'm so proud of you. Thank you very much, Prof. Colleagues, now what is going to, I'm not sure, you said you thank the directorate. You should thank me separately as a program director. Thank you. Yes. Okay. I'm just joking, colleagues. Yes, the program director, and you should explain how I'm directing the program successfully. I'm joking to you, Prof. Okay, colleagues. Here is what going to happen. We are going to all stand up and sing the national anthem, and then after the national anthem, we are going to allow that song to come, the one that we came with. So we're gonna go out as a procession again, and then after that one, everybody, colleagues, there is a nice lunch outside, and everybody is invited. Actually, you must eat until you are full, because there is plenty outside. So on that one, I want to thank God for giving us this day a success, Prof, because we have prayed for it under difficult times, but we managed. Thank you very much, everybody. Can we stand up and sing the national anthem? Who's leading us? Yes.